Let's begin first by looking in detail at the histology of the trachea. As we said, it's a hollow tube. It's about 10 centimeters in length and about 2 centimeters in diameter. It extends from the lower portion of the larynx, beginning at the cricoid cartilage, to its bifurcation at the carina to form a primary bronchus for each lung. And again, its most striking histologic feature, making it easy to identify, is its C-shaped array of hyaline cartilage rings. The rings partially overlap in the anterior part of the trachea, but their free posterior ends are separated and interconnected by smooth muscle cells. Histologically, we show a view of a portion of the trachea on the next picture. The arrow indicates the location of the hyaline cartilage ring, and toward the interior of that we see a lamina propria with seromucous glands and the classic lining pseudostratified ciliated epithelium. At higher magnification, we can see the epithelial cell types that make up this epithelium. Again, it is pseudostratified in the sense that all of the cell types in the epithelium are lining or found on the basement membrane, but only certain of the cell types extend to the luminal surface. Those cell types that extend from base to apex are the ciliated columnar absorptive epithelial cells that have prominent amounts of not only microvilli, but true cilia. The true cilia are indicated by the arrow in this particular picture. Interspersed among the columnar ciliated epithelial cells are goblet cells, indicated by the arrowhead. Additional cell types in this epithelium will be basal cells that have rounded nuclei in a cytoplasm that's anchored to the underlying basement membrane. Those basal cells are important because they act as the stem cells for the goblet cells and the columnar epithelial cells. But intermixed among the basal cells will be different populations of endocrine-producing cells as well. So this list of epithelial cell types is summarized on the next slide. We note that, again, the dominant cell types in the pseudostratified epithelium of the conduction portion of the respiratory system are the columnar epithelial cells and the goblet cells. Both extend from the basement membrane to the luminal surface. We know that the columnar cells are typically ciliated, and these true cilia are intermingled with microvilli. The presence of the cilia are essential to moving mucus and whatever's trapped in it out of the respiratory system, so the mucosal components can be swallowed at the level of the pharynx. The goblet cells secrete a polysaccharide mucus material into the lumen. It's supplemented by the secretions of the submucosal mixed seromucous glands. And again, this mucus secretion plus that of the glands provides a mucosal layer that, as we said, is moved out of the respiratory system by ciliary beat. Down at the bottom, we said that the basal cells are also an epithelial component of the pseudostratified epithelium. They only lie on the basement membrane, but as we said, they serve as stem cells for the goblet and the columnar epithelial cells. And intermingled at basal layers are various populations of pulmonary neuroendocrine or Kolchitsky cells. These are comparable to the endocrine cells elsewhere, particularly those in the gut, and they are hormone-producing cells that produce hormones, active peptides, bombesin, which is gastrin-releasing factor, leukenkephalin, serotonin, and somatostatin. If mucosal clearance is ineffective, or if the mechanism is overwhelmed, basically the infection caused by the pathogenic bacteria that are trapped in it, or any related forms of dust-related diseases may result. In cystic fibrosis, there is a defect in the chloride transport mechanism that results in the secreted mucus being thick or excessively viscous. And as a result, the cilia have a difficult time moving it out of the conduction portion of the respiratory system. Patients with this disease are obviously prone to frequent infections of the respiratory system. And as we noted earlier in our epithelial chapter, patients that have a ciliary defect in the ATPase motor molecule dynein have immotile cilia or Cartaginer syndrome. Those with this syndrome are subject again to respiratory problems because now the cilia, because they're immotile, cannot move the mucus layer effectively out of the respiratory system with its trapped bacteria. And certainly males that have this syndrome are going to have immotile sperm as well. Note again that the goblet cells and the columnar cells are particularly sensitive to irritation. The goblet cells under appropriate stimulation become taller, and there's an increase in the number of goblet cells and in the submucosal glands. Intensive irritation typically arises from smoking. It can result in a squamous metaplasia, where the ciliary epithelium becomes modified to form a more protective squamous epithelium. But fortunately, this process is reversible. And classically, bronchial metastatic tumors tend to arise from different forms of the Kulczycki or the neuroendocrine cells that are characteristic of the pseudostratified epithelium.